Hi students, Doc Ian here. So this will be the part 1 of our discussion on the nucleus of the cell. This is the picture of a nucleus. So I want you to imagine that we took out the nucleus from the cell. So the spherical structure that you can see in the photo is the nucleus. These are the parts of the nucleus. This is its nuclear envelope or nuclear membrane. Appreciate that there are 2 layers in the nuclear envelope. Next, the spherical structure that you can find in the center of the nucleus, that is the nucleolus. And the thread-like structures that you can see inside the nucleus, that is the chromatin. This is how the nucleus would appear under electron microscope. So let's try to identify the parts. The red arrow is pointing to the nuclear envelope or nuclear membrane. The blue arrow is pointing to the chromatin. The green arrow is pointing to the nucleolus. And the space where the nucleolus and the chromatin are suspended, you call that one as nucleoplasm. This is how the cells will look like under bright field microscope. And I want you to focus on the nuclei of the cells. This is the nuclear envelope. This is the chromatin. And this is the nucleolus. And the space where the nucleolus and the chromatin are suspended, you call that as the nucleoplasm. As mentioned a while ago, the nuclear envelope has 2 membranes. We will name them as outer and inner membranes. And these outer and inner membranes are separated by a space. And that space is called perinuclear space. The term peri would mean surrounding. The space is actually surrounding the nucleus. That's why it's called perinuclear space. This is a closer look at the nuclear envelope. As you can see, it has the outer membrane, it has the inner membrane, and they are separated by the perinuclear space. The organelle that you can find immediately beside the nucleus is the rough endoplasmic reticulum. It is easily identified by the presence of the ribosomes. Please take note that the outer membrane of the nuclear envelope is continuous with the rough endoplasmic reticulum, just like what is being shown by the red arrows. Another important thing to take note of, the outer membrane of the nuclear envelope is also provided with ribosomes, so that once the messenger RNA will go out of the nucleus, they can be translated in one of these ribosomes. This is the rough endoplasmic reticulum. Please appreciate that it is directly continuous with the outer nuclear membrane. Then take a look at this thread-like structure, that is the chromatin. And I want you to appreciate that the chromatin is attached to the inner nuclear membrane. And what is causing it to be linked or attached to the inner nuclear membrane are those yellow-colored figures, blue-colored figures, and even those pink-colored thread-like structures. Those are proteins responsible for anchoring, attaching the chromatin to the inner nuclear membrane. And those group of proteins we will call as nuclear lamina. So if you will be asked in the exam, what portion of the nuclear envelope is associated with nuclear lamina, the answer is the inner nuclear membrane. And if you will be asked what's the function of the nuclear lamina, it functions to attach, anchor the chromatin to the inner nuclear membrane. So this one is our nuclear lamina. Again, the inner nuclear membrane is associated with group of proteins we call the nuclear lamina. And the main purpose of the nuclear lamina is to link the chromatin to the inner nuclear membrane. Let's apply what you have learned so far. The yellow-green colored figure in the photo is the chromatin. And then you have there the blue and golden colored nuclear lamina. And please appreciate in the picture that the nuclear lamina is found between the inner nuclear membrane and the chromatin because it functions to link the chromatin to the inner nuclear membrane. Another important structure that you can find in the nuclear envelope is the nuclear pore complex, and these are the ones pointed by the red arrows. These structures function to allow substances to go in and out of the cell. By the way, the protein composition of the nuclear pore complex is called nucleoporins. Take note, our DNA is enclosed within our nucleus, and the main purpose of which is to protect the DNA from any chemical that can cause it to mutate. But the structures within the nucleus still need substances that can be found outside the cytoplasm. That's why the nuclear envelope should be provided with nuclear pores. But these nuclear pores must be strict in allowing substances to go in and out of the nucleus. It has to be selective. Why? Because not all of the substances that will try to enter the nucleus is beneficial to the nucleus. Some of them might cause the DNA to mutate. Anything small is not threatening, but something big is worrisome. That's why the nuclear pore will allow small substances to just easily go in and out of the nucleus through simple diffusion, because small substances have less tendency to cause the DNA to have mutations. Again, small substances may pass in and out of the nucleus through the nuclear pore complex through the process of simple diffusion. In terms of large substances, the nuclear pore must regulate their entry into the nucleus because some of them can cause damage or harm to the DNA. But not all large substances are harmful to the DNA. Some of them are needed inside the nucleus. So there must be a way on how the nuclear pore should choose what large substance. Most of the time, by the way, large substances are proteins. So which proteins will be allowed to go in of the nucleus and which will not be allowed to go in? The answer to that is the large substance or the protein must have in its structure what we call nuclear localization signal. Please recall that proteins are produced in the rough endoplasmic reticulum or ribosomes, but they are initially produced as immature or non-functional proteins. These proteins must be transported first to the Golgi apparatus. The latter functions to modify the proteins and decide where to transport them after. In terms of the proteins that are needed by the nucleus, the Golgi apparatus will modify them by incorporating in their structure nuclear localization signal, and the presence of which will allow the protein to go inside the nuclear pore, then into the nucleus. What protein do you think will identify if the protein has nuclear localization signal? That protein is importin. So once the importin will detect the presence of nuclear localization signal in the structure of the protein, this importin will accompany the protein towards the nuclear pore so that the protein can enter the nucleus. I want you to imagine that you are the student and you want to enter the MSF. So in the case of nucleus and the protein, you are the protein and the MSF is the nucleus. Before you will be allowed to enter the MSF, you must have your DMSF school ID. So that DMSF school ID is your nuclear localization signal. Who will identify whether you have the school ID or the nuclear localization signal? The security guard. The security guard will ask you, where's your ID? If you will be able to show him your ID, then the guard will accompany you towards the gate of the MSF. The guard in the story is acting as the importin, and the DMSF gate is the nuclear pore. Please take a look at this schematic diagram. You have here a protein with nuclear localization signal. The nuclear localization signal is the one pointed by the black arrow. So it would only mean that this protein is destined to go inside the nucleus. That nuclear localization signal will be recognized by importin. Please appreciate that the importin has two subunits. You have the alpha and the beta subunit. The subunit that will bind to the nuclear localization signal is the alpha subunit. This importin will accompany now the protein with the nuclear localization signal towards the nuclear pore. But before the complex will be able to enter the nuclear pore, the beta subunit will dissociate. And what will only go through the nuclear pore are the protein with the nuclear localization signal and the alpha subunit. Once inside the nucleus, the protein and the alpha subunit of the importin will interact with RAN-GTP as shown in the picture. The interaction between the protein and the alpha subunit of importin and the RAN-GTP will cause now the alpha subunit of the importin to dissociate from the protein, and that alpha subunit will go out of the nucleus and will pair again with the beta subunit. But at least at this point, the protein has already entered the nucleus. So take a look at this schematic diagram. You have here the protein with the nuclear localization signal. It is recognized by the importin. The main role of the importin is to transport the protein with the nuclear localization signal through the nuclear pore, then into the nucleus. After their entry into the nucleus, they will encounter RAN-GTP, and the RAN-GTP now will cause the alpha subunit of the importin to dissociate from the protein, allowing the protein now to be liberated inside the nucleoplasm. So I want you to fill in the blanks. Large proteins that are destined to go inside the nucleus need what before they can enter the nuclear pore? Nuclear localization signal. Some books would call it as nuclear localization sequence. The NLS now will be recognized by importin, which guides the protein to the nuclear pore. Once inside, the protein complex, meaning the protein and the importin, will be bound to, yes, RAN-GTP, which will cause now the importin, or to be specific, the alpha subunit of the importin, to dissociate from the protein. I want you to imagine that you will kill a person and you're going to harvest the DNA of that person. If we are going to connect all the DNA of one person, that DNA would go from Earth to Moon back and forth 6,000 times. That's how long the DNA of one person. So the next question now is, how can you fit that very long DNA into our human body? To answer that question, I want you to imagine that you bought a long wire cord, and when you placed the wire cord on the floor, it looked messy. So we need to organize the wire cord so that we can put it into a small area in your room. So what we will do is, we will roll the cord on a storage reel so that the cord will be concentrated in one area, it can be condensed on one area, and you can now organize the cord. So if we can roll the wire cord on a storage reel so that we can contain it in one area, that is also the same mechanism on how the body will also contain our DNA in that small space we call the nucleus. Please appreciate the double-stranded DNA in the picture, and then take a look at the structures where this DNA will wrap around itself onto. Those circular structures there are histones. If DNA will wrap around histones, it becomes condensed, it becomes shorter, then we can now place the DNA inside our nuclei. Take note from the name itself, deoxyribonucleic acid, expect that DNA is acidic and it is negatively charged, while histones are basic proteins, and you know very well opposites attract. Histones are positively charged, so it would be easy for the histones to interact with the DNA. The blue thread-like structure is the DNA, and appreciate that it is wrapped around the green spherical histones. What do you notice? As we wrap DNA around histones, it becomes shorter, and we condense it and make it fit into a small space or area. This is our double-stranded DNA, and our DNA is wrapped around histones. Then, as you further condense the wrapping of DNA around the histones, you will now be able to form the chromatin fiber. Then, if these chromatin fibers will be further condensed, that's the time we will be able to form what we call the chromosome. That's the reason why, when I showed you this picture a while ago, chromatin was represented by a thread-like structure because that is how the DNA would look like if we condense it around the histones. So, from now on, I want you to think that the chromosomes and the chromatins are condensed forms of DNA, and DNA was condensed because DNA was wrapped around histones. So take a look at the picture of the chromosome on the left side of the screen. As we unwind the chromosome, we get to see the chromatin. As we unwind the chromatin further, we now get to see the long strand of DNA that is wrapped around this group of circular structures which we call the histones. This is the short region of the DNA, and if we will wrap around DNA on histones, we will be able to form what we call the beads on a string form of chromatin, and that is called as a nucleosome. Please take note, as the DNA is wrapped around histones, they will form what we call beads on a string. Why beads on a string? Because the circular structure they're representing histone is the beads, and you have the DNA that is still free, that is the string, and then after the DNA you will have another histone that is another bead, and then you have another free or liberated DNA that is now the string. That's why the nucleosome is described to have a beads on a string appearance. This is how the nucleosome will look like. Take a look at the double-stranded DNA. Please appreciate that the DNA will wrap around 8 histone molecules. These 8 histone molecules will be referred to as octamere. And then, the DNA will again wrap around another group of 8 histone molecules. This is the nucleosome, and it is described to have beads on a string appearance. Why? This one looks like the bead, this one is the string, and this is another bead. That's why nucleosome is described to have beads on a string appearance. So the nucleosome is composed of DNA and 8 molecules of histones, which we will refer to as octamere. And I want you to count how many loops of DNA are formed every time the DNA will wrap around 8 molecules of histones. There are how many loops? 2 loops. So a nucleosome is composed of 2 loops of DNA, and the loops of DNA are wrapping around 8 histones molecules, which we'll refer to as the octamere. The nucleosome is said to have a beads on a string appearance. This is again another picture of the nucleosome. Please appreciate the beads on a string appearance. What I want you to learn at this point is that the diameter of one nucleosome is 10 nanometer. Again, the diameter of one nucleosome is 10 nanometer. So let's have a review. You have there on top the DNA molecule. Once the DNA molecule will wrap around 8 histone molecules, you will have the nucleosome, which has the beads on a string appearance. Then, if you will further condense that, you will be able to form what we call 30 nanometer chromatin fiber, or just simply 30 nanometer chromatin. So how do we form the 30 nanometer chromatin? The 30 nanometer chromatin fiber is composed of 6 nucleosomes. Please picture in your mind the beads on a string appearance of the nucleosome. Imagine 6 beads or 6 nucleosomes. If you will condense these 6 nucleosomes, they will be able to form the 30 chromatin fiber as shown or pointed by the blue arrow. So how do we form this 30 nanometer chromatin fiber? Take a look at the next PPT slide. This is how we form the 30 nanometer chromatin beads. So you get 6 nucleosomes, then you make them rotate on each other, then you will now have a 30 nanometer chromatin bead. These are 6 nucleosomes. Please appreciate the beads on a string appearance. So how can we make these 6 nucleosomes form the 30 nanometer chromatin fiber? We'll just simply condense them with each other. And when we condense them with each other, they will now form the 30 nanometer chromatin fiber. And this is how they will look like. Please appreciate the 6 nucleosomes in 130 nanometer chromatin fiber. So why do you think this one is called the 30 nanometer chromatin fiber? Please take a look at the yellow star. That's one nucleosome. And one nucleosome is already 10 nanometer. So if we'll measure the diameter from the yellow star down to the white star, we will be going through 3 nucleosomes. And when you add their diameters together, you will be able to come up with 30 nanometer. So that's why it's called 30 nanometer chromatin fiber. So let's have a review. This is your double-stranded DNA. The white arrow is pointing to the nucleosome, which is formed when the DNA is wrapped around octameres, the 8 histone molecules. And then when you group 6 nucleosomes together, you would be able to form what we call 30 nanometer chromatin. Take note, that is how the 30 nanometer chromatin fiber will look like from its lateral view. But if you're going to view it on its frontal side, this is how it will look like. So, this second yellow arrow is pointing to another 30 nanometer chromatin fiber. And the third yellow arrow is pointing to another 30 nanometer chromatin fiber. DNA is in the form of chromatin in metabolically active cells. But if these cells will undergo cellular division, the chromatin will be further condensed. And when they do, they will now be able to form what we call the chromosomes. So, I want you to appreciate that chromosomes are actually composed of your DNA molecules that are tightly wrapped around or condensed around histones. So from now on, every time you will see a chromosome, I want you to imagine it is the condensed form of chromatin. Chromatin, on the other hand, is composed of DNA that is tightly wrapped around histones. That's why if you can see in the photo, if you will unwind a chromosome, you will see the long strand of DNA and the molecules of histones. Take note, if the cells are metabolically active, they are not undergoing any form of cellular division, the DNA must be in the chromatin form. And if the cell should undergo mitosis, the DNA should be condensed to form the chromosomes. So what you should expect to see in a metabolically active cell is the chromatin, and what you're supposed to see in a cell that is supposed to undergo the cell division is the chromosome. Question: which among these two do you think would appear darker under the microscope? Is it the chromatin or the chromosome? The answer is chromosome, because it is the condensed form of DNA. We have here cells A and B. I want you to focus on the nuclei of these two cells. Cell A is metabolically active. Cell B is undergoing mitosis. Please appreciate that the nucleus of cell B appears darker than that of cell A because all of the DNA in the nucleus of cell B are condensed to form the chromosomes. So that would be our clue that cell B is currently undergoing cellular division. The next question is, which among these two cells has the DNA that is transcribed to form the messenger RNA to initiate protein synthesis. Please take note that for DNA to be transcribed, the two strands should be separated so that one strand can be copied to form the mRNA. And you cannot do that on a DNA that is tightly coiled around histones. The DNA must be loosely attached to the histone so that it would be easy for us to separate the two strands and copy one of the strands to form the mRNA. So the answer is cell A or cell B? It's cell A. Because cell A is metabolically active and part of the metabolism is to produce proteins. So chromatin is composed of DNA wrapped around histones, and chromatin is found in cells that are metabolically active. Chromosome is basically a chromatin that has undergone further condensation because the cell has to undergo cell division. Do you know that all cells in the body have the same DNA content? So the question now is, if they all have the same DNA content, how come these cells do not look the same? How come these cells differ in their function? Do you know that the DNA that we have on our faces are the same DNA that we have on our butts? If that's the case, your face should look like your butt and your butt should look like your face. But how come the cells in the body have different morphologies? They have different functions, given that they have the same DNA. For you to understand that, I want you all to appreciate that there are small areas in the DNA which we call the genes. And these genes are the ones responsible for the encoding of proteins. This is the DNA, and the small areas within the DNA are called the genes. This gene encodes for albumin. This gene encodes for collagen. This gene encodes for transferrin, while this gene encodes for elastin. Take note, the cells in the body have the same DNA, so expect that they have the same sets of genes. Let's take that DNA and place that on a hepatocyte or liver cell. Liver cells are known to produce albumin, so the gene portion of the DNA that encodes for albumin should not be wrapped around histones because that portion of the DNA should always be transcribed to form the messenger RNA. So, in the picture, you can see that that particular portion of the DNA remains free, and that's the string. Collagen is not needed and it's not produced in the liver cells. So therefore, that gene portion of the DNA can be wrapped around histone. Transferrin is a protein produced by the liver cells. So that portion of the DNA should remain free and not wrapped around histones. So the pattern that you are forming right now is string, bead, string. And since elastin is not needed by the liver cells and it's not also produced in the liver cells, the gene portion for elastin should be wrapped around histones. So for that particular DNA, the pattern in the hepatocyte is string, bead, free, wrapped around histones, free, wrapped around histones. So if this is the nucleus of the hepatocyte, any light areas would mean that the DNA there is not wrapped around histone. So meaning the DNA there is actively being transcribed. So any light area could be representing the gene for albumin. And the dark staining areas represent the portions of the DNA that are not needed by the cell. That's why it is wrapped around histone. That's why the dark staining area there can be representing the collagen gene. This light area can be representing the transferrin, which is also produced in the liver, and another dark area could represent the elastin. So let's just assume that these are the liver cells. So this dark staining area here is representing the collagen gene and the elastin gene, while this light staining area here is representing the genes for albumin and transferrin. Let's put the same DNA to the fibroblast of the skin. Albumin is not produced by fibroblasts. So therefore, that gene portion of the DNA should be wrapped around histone. Please recall that this portion of the DNA in the hepatocyte remains free because it is actively transcribed. But in the fibroblast of the skin, that portion of the DNA is tightly coiled. Whenever there is injury, the fibroblasts will form the scars by depositing collagen. Therefore, the gene that encodes for collagen should remain free, not wrapped around histones in the nucleus of the fibroblast. So if in the hepatocyte, that portion of the gene is tightly coiled around histones, in the case of fibroblasts, it would remain free. Transferrin is not produced by the fibroblast, therefore that gene should be wrapped around histones, while elastin is needed to form the elastic fibers. So therefore the fibroblast will also not allow that portion of the DNA to wrap around histones. So if the pattern of the DNA in the hepatocytes is string, bead, the same DNA will have this pattern in the fibroblast of the skin. Bead, string, bead, string. So if this is the nucleus of the fibroblast in the skin, any dark area now could represent albumin, while in the hepatocyte, albumin is represented by the light staining areas. So this dark staining area is albumin. This light staining area is collagen. This dark staining area is transferrin, and the other light staining areas could be elastin. And that's the reason why cells in the body, even if they have the same DNA, they differ in their morphologies and functions because they have different portions of the genes that they are actively transcribing and different portions of the DNA that they tightly coil around histones. That leads us now to the two forms of chromatin, the dark staining heterochromatin and the light staining euchromatin. Please take note that the portion of the DNA that is actively transcribed should not be tightly coiled around histone. So therefore, that portion of the DNA will appear lighter and we refer to it as euchromatin. While the portion of the DNA that is not needed by the cell should be tightly coiled around histone, therefore it will be condensed and it will appear darker than euchromatin, and that is the heterochromatin. If the cell you're looking at is known to produce proteins, expect that the cell would have higher amounts of euchromatin compared to heterochromatin. So expect that the nucleus of that particular cell should appear lighter compared to the nucleus of another cell that is not active or that does not function to produce protein. So if I will ask you in the exam, true or false, the nuclei of cells that are active in protein synthesis appear lighter than the nuclei of cells that do not function to produce proteins, the answer is true. So let's try to identify from this nucleus the heterochromatin and the euchromatin. But first, let us identify that this is the nucleolus. The dark staining area within the nucleoplasm is the heterochromatin, and found between the heterochromatin are the light staining areas which represent our euchromatin. For you to easily remember that euchromatin is needed by the cell and it's actively transcribed by the cell, you have to simply remember, I needed you. That brings us now to the different types of heterochromatin, or the dark staining forms or condensed forms of DNA inside the nucleus. The dark staining areas that are found near the nuclear membrane, particularly the inner nuclear membrane, will be called the marginal chromatin. While the chromatin that you can find associated with the nucleolus will be called nucleolar associated chromatin. The other heterochromatin that you can find between the marginal and the nucleolar associated chromatin will now be referred to as the karyosome. So this is one karyosome, this is another karyosome. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. Please understand that females only need one of their X to be active. That's why one of the X chromosomes of female should be inactivated. And as you have learned a while ago, if DNA is not needed by the cell, it's supposed to be tightly coiled around DNA. So in the case of the 2 X chromosomes of females, since only one is needed, one should be tightly coiled around histones, should be further condensed, and that X chromosome will become heterochromatin. While the active X chromosome should never be tightly condensed. Why? Because it is actively used, transcribed by the female cells, no, for them to produce proteins. So that particular X chromosome should exist as euchromatin. So expect that in the cells of females, they should have an extra number of heterochromatin. And that extra heterochromatin is representing now the heterochromatin form of their X chromosomes. Recall that in males, we only have X chromosome and Y chromosome. And we need the expression of the genes from both of these chromosomes. So we will never form a heterochromatin of our X chromosome. The heterochromatin form of X chromosome that is only present in the cells of female is referred to as the sex chromatin or the bar body. For you to understand what am I trying to say, please take a look at these 2 neutrophils. The neutrophil on the left side of the screen is from a male patient, while the neutrophil on the right side of the screen was taken from the blood of a female. Appreciate the structure that is encircled, and you don't see the same structure on the nucleus of the neutrophil of the male patient. That extra heterochromatin represents now the heterochromatin of form of X chromosome that is only present in females, and that is now the sex chromatin or the bar body. And the bar body is utilized in the laboratory to identify if the tissue or the cell that is being examined was sampled from a male or a female. So take a look at this neutrophil. Can you appreciate that you have here a bar body? So from that information, you will now know that the blood smear that you're looking at was taken or sampled from a female patient. So in the future, if you will encounter a female patient and she will ask you to examine her peripheral blood smear, and then you will notice that her neutrophils do not have bar bodies or sex chromatin, you will have an idea of the secret of that patient. Charan! The next structure that we can find within the nucleus is the nucleolus. It's the spherical structure that we can find inside the nucleus. Nucleolus is a spherical, highly basophilic structure that we can find inside the nuclei of our cells. When you say basophilic, they are attracted to basic dyes such as hematoxylin. So expect that the nucleolus will have what color? Dark blue. What's the purpose of the nucleolus? The nucleolus functions to synthesize ribosomal RNA. And this ribosomal RNA is important in the formation of the ribosomes. What's the function of the ribosomes again? The ribosome will interact with the messenger RNA. The messenger RNA code is translated to form proteins. So take a look at this picture. You have here the black-colored messenger RNA that is being translated by the green-colored subunits of your ribosome. Every code in the mRNA will be read by the ribosome, and the code will be translated to a particular amino acid. That's why you can see here a growing peptide chain. And when you say peptide chain, it's supposed to be composed of what? Amino acids linked together by peptide bonds. So the peptide chain is the one that is colored dark blue in the picture. Now, what brings the amino acid to the ribosome so that the amino acid will be added to the growing polypeptide chain is another type of RNA, and that's the transfer RNA. So the RNA that is translated by the ribosome to synthesize the proteins is mRNA. But what will load or transport or carry the amino acid towards the ribosomes so that the growing polypeptide will be assembled is the transfer RNA. Nucleolus functions to synthesize ribosomal RNA. So, what is now the role of ribosomal RNA in protein synthesis? I want you all to listen very carefully. For ribosomes to be formed, you must combine ribosomal RNA plus proteins. The ribosomes that you saw on the previous slide were assembled by the combination of ribosomal RNA plus proteins. So meaning, we can never form these ribosomes without the ribosomal RNA. And ribosomal RNA is produced by the nucleolus inside the nucleus. So I want you to imagine the DNA will be transcribed to form mRNA. While the RNA is going out of the nucleus, what will assemble the ribosome so that the mRNA will be translated later on by the ribosome is the nucleolus, because the nucleolus will produce the ribosomal RNA. So take a look at this picture. This is the nucleolus, and it functions to produce ribosomal RNA. And this ribosomal RNA will combine with proteins, and the combination of these two will now form the ribosomes. Let's now go to cell divisions. There are two types. We have so these are just few of the cells in our body. Take note, the germ cells are the egg cell and the sperm cell, and they undergo cell division in the process called meiosis, while the rest of the cells that you can find in the picture will undergo cell division in the process called mitosis. Take note that for somatic cells, the total number of chromosomes is 46. And for each chromosome, you always have a pair. So, take a look at the picture. You have there 23 pairs of chromosomes. 23 times 2, that is equal to 46. In germ cells, such as the egg and the sperm cells, they only have half of the total number of chromosomes of somatic cells. That's why for every chromosome number there, you only have one chromosome. So let's have the following terms: diploid and haploid. Somatic cells have 46 chromosomes. Since the 46 number is twice the 23 chromosomes of our germ cells, then you will refer to somatic cells as diploid. On the other hand, germ cells only have half of the number of chromosomes of somatic cells. That's why the germ cells are referred to as haploid, for they only have 23 chromosomes. There's a reason why germ cells should only have half of the total number of chromosomes of somatic cells. So take a look at the chromosomal makeup of the egg cell on the left side of the screen and chromosomal makeup of the sperm cell on the right side of the screen. They both have 23 chromosomes each. But if the sperm cell will fertilize the egg cell, their chromosomes will combine together and they will now form an individual with a total number of 46 chromosomes. So for every chromosome number there, you have a pair. One came from the egg cell, mother. One came from the sperm cell, father. That's why I want you to understand that for every chromosome number in there, one was inherited from mother and the other one was inherited from father. So, humans have a total of 46 chromosomes, 23 of which came from the father, while the other 23 came from the mother. So take a look at the last pair of chromosomes. Please appreciate that they are labeled X and Y. So that will give us an idea that all of these chromosomes were taken from a male person. So the last pair of chromosomes are referred to as sex chromosomes because it will give us an idea of the sex or gender of the patient. Again, the last pair of chromosomes is referred to as the sex chromosome, and it can be a combination of two X chromosomes or one X and one Y chromosome. The first 22 pairs are referred to as autosomes. At this point, we'll just get one of the pairs of chromosomes. So let's say, for example, let's get chromosome number 8. So this is chromosome number 8. You know very well that one was inherited from the mother, while the other one was inherited from the father. This is how chromosome number 8 will look like before the cell will undergo mitosis. But if the cell decides to undergo mitosis, it must produce daughter cells with the equal number and type of chromosomes. So, chromosome number 8 must be doubled so that once the cell will split into two daughter cells, the chromosome number 8 will be equally shared. So, I want you to appreciate in the picture how chromosome number 8 was doubled. So, expect that during mitosis, this chromosome number 8, mama, papa, will look like this one. You will have chromosome number 8, mama, mama, papa, papa. Chromosome number 8 must be duplicated so that if this cell will undergo mitosis, the daughter cells will receive the exact copies of the chromosomes. Let's go to the term chromatid. Chromatid is one half of the two identical copies of a replicated chromosome linked by a centromere. So, I want you to take a look at the picture of a single chromosome. That single chromosome is also considered to have one chromatid. But if that chromosome will be duplicated, you will now have two chromatids linked together in the center by a centromere. There's a difference in stating the numbers of chromatids and chromosomes. You identify the number of chromosomes by counting the number of centromeres. So take a look at the unpaired chromosomes on the left side of the screen. If you will be asked how many chromosomes are present, just simply count the number of centromeres. 1, 2. You do not mind how many arms are there, you just simply count the number of centromeres. Let's take a look at the duplicated chromosomes on the right side of the screen. If you will be asked how many chromosomes are present, again, just count the numbers of centromere. So for the duplicated chromosomes, you still have 2 chromosomes because basically you only see 2 centromeres. If you will be asked how many chromatids are there, you should count the number of arms. So for the unduplicated chromosome, you will count the number of arms, and you can see 2 arms. So you will report the unpaired chromosome to have 2 chromosomes and 2 chromatids. 2 chromosomes because you only base it on the number of centromeres. Let's go to the duplicated chromosomes. They have total number of 2 chromosomes because they only have 2 centromeres. But if you will be asked how many chromatids are present in there, you have to count the number of arms. So how many chromatids can you find on the duplicated chromosomes? You have 4 arms, and that is now equivalent to 4 chromatids. At this point, we will be talking about the cell cycle. This is basically the life story of the cell. Please take note, it is not all the time that the cells are dividing because they need to allot some time doing their function. That's why in reality, the cell would only spend 10% of its total lifespan in the mitosis stage or the actual cell division, while it will spend 90% of its time on G1, S, and G2. These G1, S, and G2 are stages of what we call interphase. The first stage in interphase is G1. The letter G there stands for the word gap because interphase serves as a gap between 2 successive mitoses. Now, what's the importance about G1 phase? As you can see in the picture, it's the step over or the stage of interphase that immediately follows the process of mitosis. So at this point, you have the production of the daughter cells from the mother cell. So expect that in this stage, the cell will perform its function. The cell should be metabolically active. And since it is a new cell, it's supposed to be growing in size. And I want you to appreciate in the picture that what will follow G1 is S. S stands for synthetic phase. Why do we call this stage as synthetic phase? Because it is at that stage that the cell will duplicate its chromosome or DNA in preparation for another round of mitosis. So, to have the energy to duplicate its DNA or chromosomes, the cell in the G1 must gather all the nutrients and materials needed for the DNA to be duplicated at the S phase. The cell on the left side of the screen is the mother cell. If that mother cell will undergo mitosis, it will produce 2 daughter cells, and these 2 daughter cells will enter now the G1 phase of the interphase. Expect that on the G1 phase of the interphase, all of the chromosomes should be unpaired. But as mentioned a while ago, once the cell will reach the synthetic phase, the DNA and the chromosome should be duplicated. And this is how they will look like. And since they have already been duplicated during the S phase, so expect that the chromosomes will also be duplicated when the cell reaches the G2 phase or the gap 2 phase. At this point, we will try to count the number of chromatids and chromosomes in each of the stages of interphase. Let's start with G1 phase. How many chromosomes can you find in the picture? 2. So if you're going to combine this chromosome with the other chromosomes, you're supposed to have how many? 46 chromosomes, right? Since the chromosomes will be duplicated in S phase, this is how it will look like. But since we base the number of chromosomes on the number of centromeres, then the total number of chromosomes in the synthetic phase will still be 46. Because the number of the centromere did not change. What changed is the number of the arms. So for G1 phase, you have 46 chromosomes. So it follows, since they have only 1 arm per centromere, you have 46 chromatids. But as soon as the cell will enter the S phase, the number of chromosomes will still be the same, 46 chromosome, but since the arms are duplicated, expect that the number of chromatids at the S phase will be 92. So the number of chromosomes and chromatids will be the same between G2 and S. What I want you to learn from this particular slide is that cells do not just simply jump from G1 to S because the cells must go through what we call checkpoint in the cell cycle. So meaning, it's not easy for the cell to just say, okay, I'm ready to duplicate my DNA, then therefore I will go to S phase. They need to meet some of the requirements which will be checked by the checkpoints in the cell cycle. So what are the checkpoints present between GAP1 phase or G1 phase and S phase? These are the checkpoints. You have restriction point and G1 DNA damage checkpoint. Take a look at the picture of the life cycle, and I want you to focus on the area between the GAP1 phase and the synthetic phase. You have here one of the checkpoints, which is restriction point, and you have the second one, which is the G1s DNA damage checkpoint. What is now the purpose of these two checkpoints? Let's try to figure out. The restriction checkpoint allows the cell to evaluate its potential to replicate. So the cell will be asking itself at this point, do I have enough nutrients for me to be able to duplicate my DNA? Do I have enough of the growth factors so that I can push through with duplicating my DNA? If the cells will decide that they can no longer duplicate their DNA because they don't have the capacity, they don't have enough nutrients and growth factors, these cells will not proceed to S phase. Instead, they will go out of the cell cycle and go to GO. I want you to look for the GO in the picture. GO. O there stands for out of the cell cycle. Since they cannot anymore duplicate their DNA and they cannot anymore undergo another round of mitosis, they will decide to retire. And they retire quote unquote, by going to the GO stage or phase. So for you to understand what I'm talking about, take a look at the G1 phase of the interphase. Appreciate the area labeled as G1 checkpoint. That is referring to the restriction point. That is where the cell will ask itself, do I have the size to undergo another round of mitosis? Do I have the nutrients and growth factors to make me duplicate my DNA so that I can also survive through the 4 stages of mitosis? That is now the restriction point. And if the cell will not pass the restriction point, then the cell will go out of the cell cycle and stay on the resting state, and you call that as the GO phase. O means out of the cell cycle. The other checkpoint that we can find between G1 and S is G1s DNA damage checkpoint. From the name itself, you know that at this particular checkpoint, the DNA has to be checked for its integrity and presence of mutation or damage. You should not allow the damaged or mutated DNA to be replicated in the synthetic phase. That's why you must have a checkpoint that will check the integrity and the quality of the DNA. Or else, mitosis will produce 2 daughter cells with abnormal or mutated DNA. G1 phase will be followed by the S phase, the synthetic phase. It is in this particular stage of interphase that DNA replication happens. Expect that the number of chromosomes will still be the same because we will base it on the number of centromeres, but expect that the chromatids will be doubled. That's why at this particular phase, you have a total of 46 chromosomes, but with 92 chromatids. You have learned so far that before a cell will progress from gap1 phase to S phase, you have two checkpoints. After the cell will duplicate its DNA in the synthetic phase, the cell will go to gap2 phase or G2 phase. And that's the last stage of interphase before the cell will undergo the actual cell division, which is the mitosis. But take note, before the cell is allowed to progress from G2 phase to mitosis, the cell will again encounter checkpoints. What are these checkpoints? We'll discuss them in the succeeding slides. Gap2 phase or G2 phase is the last stage of interphase prior to the actual process of mitosis. So expect na at this stage, the cell will be gathering all the nutrients and energy for the actual mitosis or cell division. Now, before the cell is allowed to move from the G2 phase to the actual stage of mitosis, the cell has to encounter two checkpoints. Number 1, G2 DNA damage checkpoint. So, after the duplication of DNA in synthetic phase, the quality of DNA will be again checked by the G2 DNA damage checkpoint. And it's also very important in the G2 phase to check if all of the DNA are duplicated. You cannot afford to produce daughter cells with unequal amount of DNA or chromosomes. That's why the second checkpoint is unreplicated DNA checkpoint. So the cell has to pass through these two checkpoints before it is allowed to engage itself in the prophase of mitosis. So take a look at this picture. This is the gap2 phase, and before the cell is allowed to move to the mitosis cycle, the cell has to undergo two checkpoints. The one that will check for presence of damages and the one that will check if all of the DNA are really duplicated. If the cell will pass through these checkpoints, then the cell will now enter the actual mitosis. I hope you can recall from your previous science subjects that mitosis has 4 important stages. These are the prophase, metaphase, anaphase, and telophase. As defined a while ago, mitosis is the cellular division of somatic cells in the body. So these cells are supposed to have what? 46 chromosomes, and these cells are called diploid cells. Later on, you will learn that mitosis will produce 2 daughter cells. And since the number of chromosomes of the daughter cells are similar to the mother cell, which is a somatic cell, expect that the 2 daughter cells are diploid as well. Again, the 4 stages of mitosis are the following. They are arranged in order: PMAT, prophase, metaphase, anaphase, and telophase. The first stage of mitosis is prophase. We will be discussing the different events that will happen to the cell if the cell will enter the prophase stage of mitosis. Take a look at the red arrow. It's pointing to the nucleus of the cell. Please do appreciate that the nuclear envelope of the nucleus is starting to disappear. That's why you can see there that the nuclear envelope is starting to be broken down into pieces. Please take a look at the nucleus. Can you still appreciate the nucleolus? No, because it has also disappeared. Take a look at the chromatin. Can you appreciate that the chromatin of the cell is starting to become more condensed and they will now become visible in the form of chromosomes? I hope you can remember in our discussion a while ago, for metabolically active cells, the DNA exists as U or heterochromatin. But for a cell that is about to divide, the chromatin will condense further to form the chromosomes. So you have memorized 3 important events so far. Number 4, the centrioles or centrosomes will duplicate. That's why you can see 2 centrioles in the picture, and they will move towards the opposite sides of the cell. And these centrioles now will start forming or synthesizing the blue-colored mitotic spindles that you can see in the picture. Let's review the events associated with prophase. Nuclear envelope will disappear. Nucleolus will disappear. The chromosomes will be formed from the condensation of chromatin. You have the duplication of the centrioles and the initiation of the synthesis of mitotic spindle. So in prophase, chromosomes continue to become more condensed. That's why they will become thicker. And since they are condensed, they will become shorter. The centrioles will replicate and they will migrate towards the opposite poles of the cell. And while they are doing that, they are also synthesizing the mitotic spindles. And it's also obvious that the nuclear membrane will disappear, and as well as the nucleolus. So this animation will just show us the production of the mitotic spindle fibers by the replicated centrioles. And please appreciate in this animation the duplication of the centrioles, the production of the mitotic spindle, and the disappearance of the nuclear envelope. After the completion of the prophase, the chromosomes will then align in the center of the cell. The cell has now entered the stage we call the metaphase. So in metaphase, the chromosomes are aligned in single file arrangement in the center of the cell. These are pictures of cells that are currently at the metaphase stage of mitosis. Appreciate that the chromosomes are dark appearing because they are the condensed form of chromatin, and they are all aligned at the center of the cell. After metaphase, the sister chromatids will now be pulled towards the opposite poles by the mitotic spindles produced by the centrioles. So basically in anaphase, the sister chromatids are pulled towards the opposite sides, distributing now equal and same copies of chromosomes to the future daughter cells. Again, in anaphase, there is separation of the sister chromatids, and they will move towards the opposite poles. As soon as the chromatids will reach the opposite poles, the cell will enter the next stage of mitosis, and that is the telophase. Telophase is basically the opposite of the events in prophase. As you can see, you have here the reappearance of the nuclear envelope as shown in the picture. The chromosomes will become less condensed and they will go back to form the chromatin. The nucleoli will reappear while it disappeared in prophase. And please take note that the cell will continue to elongate. That's why you can see that the cell in this picture is somehow oval in shape. And there's a purpose for that. The cell has to elongate so that later on it can form a cleavage furrow, which will eventually cause the separation of the two daughter cells in the process called cytokinesis. Let's have a review. This is how the chromosomes of a somatic cell would look like prior to synthetic phase of interphase. This is now the chromosomes of the same cell would appear after the synthetic phase up to the metaphase stage of mitosis. What do you notice? The chromatids for each of the chromosome is doubled. I want you to imagine that these chromosomes will be arranged in a single file in the center of the cell during the metaphase stage. And you know very well that during the anaphase, the chromatids will be separated from each other and they will migrate towards the opposite pole. That's why each of the daughter cell will have the same copies of chromosomes as that of the mother or parent cell. So, let's count the number of centromeres in the photo. You can actually count 46 of them. That's why the daughter cells in mitosis are diploid. And they will all have a total of 46 chromosomes each. Let's have a summary of mitosis. Mitosis is the cellular division of somatic cells. Somatic cells are described to be diploid, meaning they have a total of 46 chromosomes. Mitosis will result to the production of two daughter cells with the same copies of chromosomes as the parent cell or the mother cell. Somatic cells, 46 chromosomes, diploid. At this point, we will now try to classify cells based on their proliferative activity. Those that no longer divide because they prefer to stay in GO or out of the cell cycle are called the static cell populations or the permanent cell population. Examples of these are our neurons, because once a person will develop stroke, you will notice that the person will no longer be able to regain the same amount of muscle strength brought about by the loss of neurons. But let me be clear with this one. Recent studies have shown that neurons can regenerate. It's just that the regeneration takes time. Another example of a static cell population is the cardiac muscle. That's why if a person had previous heart attacks or acute myocardial infarction, expect that their heart will no longer be functioning as good as before, because the remaining cardiac muscle cells cannot replicate and replace the dead ones. The next type of cells based on proliferative activity is the stable cell population or the quiescent cell. These cells have limited mitotic activity. Just like the static cells, they also stay in the GO phase of the interphase. But should there be damage or injury and there's a need to replace dead cells or missing cells, these cells are willing to move from GO back to G1 and have another round of cell cycle. Examples of stable cell population are, number 1, hepatocytes. Hepatocytes can regenerate and replace missing or dead hepatocytes or liver cells. That's why if one person will donate the right lobe of his liver, the left lobe, since it has hepatocytes, the hepatocytes there will try to regenerate and somehow the left lobe will give rise to the missing right lobe. But under normal conditions, these hepatocytes will not multiply. They will stay in the GO phase of the interphase. Another example of a stable cell population is the fibroblast in the connective tissue of our skin. If there's no injury, then they will remain inactive. Should there be a, let's say, lacerated wound, these fibroblasts will move from GO to G1. They will increase their number because with number, they can produce enough amount of collagen which will later help in the formation of the scar and facilitate tissue repair. Another example of stable cell population or quiescent cell group is the osteoblast of the bone. Once a woman will reach the age of 18 and a man will reach the age of 21, their bones will stop growing in length. But if there is a bone fracture and if the bone is allowed to heal, the osteoblasts in the bone will move from GO to G1 so that they can increase their number because osteoblasts are known to deposit bony matrix, and the deposition of the bony matrix will help in the repair of the fractured area. The third group of cells based on proliferative activity are the ones that have regular mitotic activity, and we refer to them as continuously dividing cells or labile cells. Examples of these are the blood cells. Next, the cells of the epidermis of the scalp resulting to the formation of the hair. That's why the hair will always increase in length through time. You have the epithelial cells of the skin and other organs in the body. The epithelium functions to provide protection. So it's the part of the organ that is always exposed to injurious agent. Should the cells of the epithelium die, the epithelial cells can always replace the dead ones because they can continuously divide. That leads us now to the last topic of the nucleus part 1 discussion, and that's the telomeres. Telomeres are structures that we can find at the ends of the chromosome. So those black colored structures that you can find on the tips or ends of the chromosomes are the telomeres. They function to protect the chromosomes during cell division. But as the cell will undergo cell division, these telomeres are observed to become shorter. And there will come a point that the chromosomes of that cell will no longer have telomeres, and the cell cannot anymore undergo replication. Dogs have shorter telomeres than humans. That's why they die earlier than humans. Cancer cells, on the other hand, have enzymes that can maintain the size of their telomeres. That's why cancer cells are free to replicate or multiply anytime because they will never run out of telomeres. That's why in the future, I want you all to do a research that can come up with a drug that can inhibit that particular enzyme used by cancer cells to maintain their telomeres. As the cell divides, the telomere becomes shorter until such time that the cell will no longer have telomere and the cell will become old and the cell will die. Thank you very much for listening to my lecture on the nucleus part 1. Of meiosis. Mitosis is the cell division of the somatic cells in the body, while meiosis is the cell division of the germ cells in our body. When you say germ cells, these cells are involved in reproduction. So, we're talking about the sperm and the egg cells. Cells other than the sperm and the egg are all referred to as somatic cells. So, when they divide, you refer to the cell division as mitosis.